Our dear students, welcome to lesson 7. We are looking at chemicals of life, and in this lesson we are going to introduce another chemical of life, organic chemical of life called lipids. I know you must have heard about lipids at your O level. So what do you think the lipids are? Well, probably you are aware about the fats and the oils. Yeah? So those are the lipids we are going to look at this today. And I want to welcome you. Please get your summary book and move closer so we go together so that I don't leave you behind. So when you look at the lipids, you realize that these are large group of organic compounds, just like the carbohydrates. They also contain carbon, they contain hydrogen and oxygen. But the difference is that when you look at the quantity of oxygen, the proportion of oxygen, you realize that it is smaller than in carbohydrates. Hence, they have more hydrogen compared to oxygen. So we can easily say that they are more reduced. They are more reduced, and the carbohydrates are more oxidized, for that matter. But they have the three major elements: carbon, hydrogen, and oxygen. Just like the carbohydrates. The only difference is that for them they have little oxygen compared to the carbohydrates. So let's proceed. When you look at the lipids very well, you realize that they possess some of these properties. The lipids are insoluble in water, meaning they cannot dissolve in water. You have probably tried to dissolve cooking oil in water. When you pour cooking oil in water, what happens? Usually it floats on top of the water. They are of two major types. We have fats and oils. The difference is that the fats are solid at room temperature and oils at room temperature are liquid. So that is the difference between the fats and the oils. But fats and oils are collectively called lipids. Lipids are made up of fatty acids and glycerol. You know that glycerol is an alcohol that contains 3 OH groups. Yeah, and each combines with a separate fatty acid to form a lipid that is chemically known as the triglyceride. So we're going to see how the triglyceride is formed shortly, but for now we know that when we are forming these lipids, they are formed from the building blocks called fatty acids with glycerol. So fatty acids react with glycerol to form what we call the triglyceride. Now, in this kind of reaction, that kind of reaction leads to the loss of water. So 3 water molecules are lost, and that reaction is called a condensation reaction. A reaction that involves combination of smaller molecules to form a larger molecule with loss of water is what we call a condensation reaction. So similarly, like in carbohydrates, like in disaccharides, like in polysaccharides, also in the formation of triglyceride, a condensation reaction occurs. So you can look at that reaction. From there you can see glycerol plus 3 fatty acids to form 1 glyceride molecule plus 3 water molecules, as you can see from there. So one glycerol molecule has 3 OH group, the hydroxyl groups, 3 of them, and then this allows, of course, the combination of the reaction with 3 other fatty acids, because the OH group is the most reactive part of the triglyceride. Meaning, since it has 3 OH groups, it can accommodate up to 3 fatty acids, as you can see there. 3 fatty acids, each bonded to an OH group, and then, of course, the hydrogen lost and the OH is lost. Hydrogen from the glycerol and the OH from the fatty acid forms water molecule. That's why you can see the other 3 water molecules at the product side. So you can look at that reaction critically and then try to rewrite it for yourself to see how the triglyceride is formed from a reaction between glycerol and fatty acids. So when we look at fatty acids, you realize that all occurring lipids have glycerol and fat, and therefore it is the nature of the fatty acids which determines the characteristic of any particular lipid. So in the formation of lipids, all of them have glycerol. What differs is the fatty acids, because for them there are many. Yeah, so depending on the type of fatty acid that exists in that lipid, the properties of the lipid may be different. Yeah, depending on the type of fat and example of the fatty acid present. All of fatty acids have a carboxyl group COOH. Yeah, the remainder of the molecule being a hydrocarbon, which has a varying chain. It's a hydrocarbon chain which has a varying length. So in a fatty acid, there are 2 major groups. We have a carboxyl group, COOH, which is very reactive, and then also a carbon chain, which is long or short depending on the fatty acid. These chains may possess 1 or more bonds. They may contain 1 single bond, or they may contain double bond. Yes. So if they contain a double bond or many double bonds, that acid is unsaturated. Yeah. If it possesses no double bonds, then it is said to be saturated. So therefore, fatty acids can be grouped based on the number of double bonds they possess, or whether they possess double bonds or not. When they possess double bonds, they become unsaturated because they will have less number of hydrogen atoms, so they are unsaturated. If they possess no double bond, that means they have more hydrogen atoms and therefore they are more saturated. So we have saturated fatty acids and unsaturated fatty acids. Saturated fatty acids do not have double bonds. Unsaturated fatty acids have double bonds. As you can see there, we have a number of them. As you can see, nature of fatty acid, we have butyric acid, and the general formula is there. That's 3 carbon atoms, 7 hydrogen atoms, and the COOH group, which is a functional group for fatty acids. And that one is a saturated acid, saturated fatty acid, simply because it does not contain the double bond. Yes, and is found in butter fat usually. Then we have linolenic acid, and that one has 17 carbon atoms, actually 18 carbon atoms, or we can say 17 carbon atoms, 31 hydrogen atoms, and then the COOH, which is a functional group. And that one does not have, that one has double bonds, and that one is found in seed oil and is unsaturated. What makes it unsaturated is because of having double bonds. And then we have oleic acid. We have oleic acid, which is also unsaturated, as you can see there. Palmitic acid, cerotic acid, arachidonic acid, and so on and so forth. What I wanted you to know is, how does an acid become saturated or unsaturated? Yeah, saturated fatty acids are those that do not have double bonds. Unsaturated fatty acids are those that have double bonds. So that is great for you to know. Now from that table we have seen that the hydrocarbon chains may be very long depending on the number of carbon atoms, forming very long tails which extend from the glycerol molecule. These tails are hydrophobic or water repelling, which makes the lipids insoluble in water. So as you saw in that table, you may realize some fatty acids have 17, 33 carbon atoms. So the longer the chain, the more hydrophobic it is, the more insoluble it is in water. Yeah, but generally all lipids are insoluble in water, but the longer the chain, the more insoluble it is. We have the first assignment there. Explain why lipids are insoluble in water. We can try that out, and then you send me that assignment. Try to explain that. We have already hinted about it, but you can write a short essay of about half a page or 2 paragraphs, and then you send it to me. I would want to see that short explanation of why lipids are insoluble in water. Then we have a category of lipids called phospholipids. These are lipids in which one of the fatty acid groups is replaced by the phosphoric acid. Remember, during the formation of lipids, we have 3 fatty acids and 1 glycerol molecule. So one of the 3 fatty acids is replaced by phosphoric acid. The product will be a phospholipid. The product will be a what? A phospholipid. As you can see here, we have a triglyceride here, first of all. Now, if you remove one of the fatty acid chains and then replace it with a phosphoric acid group, then you'll be forming a phospholipid. You'll be forming what? A phospholipid instead of a triglyceride. Yeah. So you can see from here, we have 2 fatty acids on your right, those ones which are highlighted, and then on your left here we have a phosphate group there from phosphoric acid. So in that case, you form a phospholipid. In that case, you form what? A phospholipid. A phospholipid, therefore, is a product of glycerol plus 2 fatty acids and then 1 phosphate group or phosphoric acid group there. So then there is another group of lipids called waxes. Probably you must have heard about waxes. These are formed by a combination of fatty acids with an alcohol other than glycerol. So when fatty acids, these ones we are talking about, combine with another alcohol which is not glycerol, the likelihood it will be wax. You're going to form wax. And their major function is they do waterproofing in surfaces of animals and plants. And there are also storage compounds in seeds, e.g. castor oil seeds. You have probably seen wax in many plant leaves. You say they have a wax cuticle. It means they contain wax which waterproofs the leaf surface. An example is the one for bananas. You can go to a banana leaf and turn it behind. You look at the lower part of the leaf. You will see some white like ash. That one there is wax. Yeah, and bananas have a high concentration of wax, and their leaves are waterproof. So they act as waterproof materials for plants and even animals. Yeah. Then we have steroids. These are related to lipids, e.g. cholesterol in animals, used to synthesize sex hormones. Other steroids include vitamin B and bile acids. Steroids are also having a closer structure with the lipids. Yeah, they have something closer with the lipids. They have almost the same structure, but they are varied. Yeah, for example, cholesterol that we know very well, which is in animals, used for formation of or synthesis of sex hormones, progesterone, testosterone, estrogen. Those are the steroid hormones we're talking about. And then also other steroids may include vitamin B and bile acids that are very vital in the bodies of mammals. So let's look at the structural functions of lipids. I want you to be summarizing them as we look at them together. The first one is that they are components of the plasma cell membrane. You remember this in your cytology, that the plasma cell membrane or the cell membrane contains of the phospholipid bilayer. Phospholipid bilayer. So basically, that phospholipid bilayer is a lipid derivative. And then, of course, we know that cell membranes are all over the body. Yeah, wherever there are cells in the body of an animal, cell membranes are there. They form subcutaneous fat in the dermis of the skin, hence insulating the body against heat loss because they are poor conductors of heat. So usually you find them under the skin, keeping the body warm, insulating the body against what? Heat loss. So those people who have more fat should be warmer than those of us who have less fat in the bodies. They are components of the wax cuticle in plants and insects, thereby preventing water loss or desiccation. We have just mentioned about fats, the wax cuticle of plants, where this wax prevents, which acts as a waterproofing material. Also, waterproofing we look at the chitin in insects. In insects, they associate with wax as well to waterproof their bodies. They form components of myelin sheath of the nerves, hence playing a role in transmission of impulses. In coordination, we looked at the structure, the myelin sheath, the structures of the neurons. You realize that there is a part called the myelin sheath, which is made up of fat, which is a very good component in the transmission of impulses. And then they protect delicate organs, e.g. the heart, kidney, from injury. So they act as shock absorbers. You've seen that around the delicate organs, there's always fat, around the heart, around the intestines. If you have eaten intestines of the goats or cows, in Luganda they say amasavu gawoma gocha, meaning it's good to eat these fats when they are still hot. When they cool, then they solidify. Yeah, they solidify. So meaning these organs have fats. The kidneys, the heart, and other organs. The intestines, those fats play a very key role of shock, absorbing shock. Yeah, and protecting these organs from damage. Also, they coat on fur of animals, enabling to repel water, which would otherwise wet the organism. So you've seen in some animals, especially those that live in water, their fur is coated with fats, with the lipids, waterproofing the animal. You've seen some birds that swim in water and they remain dry. They are not wet. Yeah, they dive in water, but their feathers remain dry. So it's because of the repelling factor of the lipids that are found in their feathers and and the fur. There are also components of adipose tissue, which is usually found under the skin. Sometimes metabolized, produce energy. Those are some of the structural functions. Let's look at the physiological functions. Functions which are related to functioning of the body are called physiological what? Functions. The first one is they provide energy through oxidation. Actually, fats produce almost 2 times the amount of energy that can be provided by carbohydrates, meaning they are a very good energy source. Secondly, they are solvents for fat-soluble vitamins. You know that vitamin A, D, E, and K do not dissolve in water, but they dissolve in fats. So the vitamins are kept in fat. And by the way, rarely do organisms suffer from deficiency of these vitamins because they can be stored in fat for long, unlike the water-soluble vitamins, which are lost always from the body. They are a good source of metabolic water to desert animals. For example, the camels, also young birds and reptiles, which are still in their egg shells. So, for these desert animals, when you look at the hump of that camel, it contains fat. And when that fat is metabolized, it produces what we call metabolic water, which is used to sustain life processes in that organism. Because in deserts, the water is always scarce. So they use the fat, they burn the fat and produce the water, which they use for other body activities. Also in organisms that that lay eggs, those eggs have some fat, and that keep those organisms, that provide them the nourishment until they hatch. They are also constituents of brown adipose tissue, which provides heat for temperature regulation or thermogenesis. We had mentioned about this earlier. Other functions of lipids include the following. Some lipids provide scent in plants, which attract insects for pollination. So you've probably seen some of the plants that have very good scent from their flowers, attracting insect pollinators. Yeah, that scent is always a derivative of a certain form of lipid. And then wax is used by bees for construction of their honeycombs, as you can see there. Also, wax can be used for production of candles and so on. That very wax that we have seen there already, wax from the bees is used in the manufacture of candles. So I want to throw this question to you. What properties do lipids possess as storage compounds? So what enables the lipids to be very good storage compounds? That's what we are asking here. So I want you to try to summarize, to write down an essay of the properties that lipids possess that enable them to become or to be good storage compounds. And then you send me those responses. I would be interested in seeing your answers there. So after looking at that, let's look at tests for lipids. So what is important here is for us to know the reagents that we use for these tests, the procedures, the possible observations, and then the possible deductions. So we have a number of tests here, but about 3 of them are good. They are possible in the laboratories. So you can do what we call the Sudan 3-dye test, and then we have the ethanol emulsion test, and then we have the translucent spot test. So we want to see what each one of them means. So to start with the first one, the Sudan 3-dye test. So the reagent, of course, is the Sudan 3-dye or the Sudan 3 reagent. The procedure is to 1 centimeter cubed of the food solution, add 1 centimeter cubed of Sudan 3 dye and shake. So if you are the one writing this for yourself, you would say that 1 centimeter cubed of food test was added, 1 centimeter cubed of Sudan 3 dye test and shaken, because you are giving a reported speech of what you did. Then the observation would be, if it is positive, if the lipids are there, a turbid solution turns to a red emulsion. A turbid solution turns to a red what? Emulsion, meaning lipids are present. Then if the turbid solution or a colorless solution remains turbid or remains colorless after shaking, then lipids are absent. So that is what we call the Sudan 3 dye test. Great. Then the other one, the second one, is the emulsion test. Sometimes the ethanol emulsion test. And our procedure would be, to 1 cubic centimeter, you write that in full, centimeter cubed, yeah? You don't write cc, we write centimeter cubed. Of a food solution, add 1 centimeter cubed of ethanol and shake. Then add 5 drops of water and then shake, continue shaking. Or if you are the one writing, you would say 1 centimeter cubed of a food solution was added, 1 centimeter cubed of ethanol and shaken, followed by 5 drops of water and shaken. Or you can add water then shake at once. No problem with that. But at least you shake with ethanol, because ethanol is a solvent for lipids. So the reason why you shake is to enable ethanol to dissolve the lipid. So the observation is a turbid solution turns to a cream emulsion. Some people would say a milk emulsion. That's okay. So whatever you see, you write. Lipids present. So if the turbid or colorless solution remains turbid or colorless, then lipids are absent. So what you are able to see here is a creamy emulsion or a milky emulsion. That is what we call the emulsion test, or the lipid emulsion test. I want us to watch this video. Our video is failing to play, but I will show you in the next lesson. Let's proceed for now. It is just explaining how we can test for lipids using ethanol emulsion reagents. And then the other one, the last one, is the translucent spot test. So the procedure here is that add 2 drops of a test solution on a piece of filter paper. This is usually done for oils. Yeah, where you add drops, those are for oils. If it is a solid like a fat, then you just rub. You rub that fat onto a filter paper. So if it is an oil, you add 2 drops of test solution on a piece of filter paper, then you allow it to dry. And then after it has dried, you observe it against light, maybe through the bulb or sunlight. So what would you see? You would see a translucent spot or a patch which is left on the paper. So you would see a permanent translucent spot on the paper, meaning lipids present. You have to add that word permanent. A permanent translucent spot will be seen on the paper, meaning lipids are present. So if there is no translucent spot, if the paper goes back to the original state, meaning no translucent spot forms, meaning lipids are absent. I told you that if it is a fat, then you just rub that fat on your paper. You don't even have to only use filter paper. You can even use any plain paper at home, and you use the cooking oil and put drops. Yeah, you can put drops on that paper, and then you dry it. And then after some time, you observe it against light, then you see whether a translucent spot has formed. If it has formed, that means your cooking oil has lipids. If the paper goes back to normal, then your cooking oil does not have lipids. The solution you have tested does not have lipids. You can do this at home and then send me a picture of what you have done. So that is what you would easily see. So if you are comparing cooking oil and water, you realize that where there was water, the filter paper remains the same, goes back to normal after drying. Where there was cooking oil, the translucent spot will be seen. I want you to do this particular test and then send me your findings. So I want to thank you very much for being very attentive and being good students. Continue researching about lipids, expound more. You can read more textbooks and find out more details about lipids and tests for lipids, so that you can familiarize with this content. Thank you very much. Stay safe.